All right, so questions over previous material? Would you like me to echo a little more? Holy crud, it's turned up to red. Apparently everyone who's in here before me like whispers. All right, so no questions? We're good? You've studied it? Yeah, in the last week when you're studying for micro and all that? Thanks, man. So, we're looking at biotin. We left off here last week, so this is back in the water-soluble vitamins chapter on page number 147. We talked through all the water-soluble vitamins, specifically B1, B2, B3, B5, and B12, and folate, which is B9, not for Dr. Sarkar's purposes. So today we're going on to B7, which is biotin, which is again B7 for the rest of the world, not for Dr. Sarkar. I don't think he disagrees with it. He just doesn't test you on that or label it as that for some reason. I don't know why. I always forget to ask him. So we're looking at biotin. Big keynote right here in the beginning. Biotin is in large portions supplied by the intestinal bacteria, which means you can get an insufficiency how, a deficiency how. You can get a deficiency through antibiotics, and we're on page 147. Yes, sir? You can get a deficiency through antibiotics for anything supplied by intestinal bacteria, so we looked at B7 supplied by intestinal bacteria. Again, great sources, don't worry about them. You'll talk about them a little bit in clinical nutrition, but not so important in here. It doesn't have this written anywhere, but the active form of biotin is carboxybiotin or biocystin. Carboxybiotin or biocystin is the active form. Like I said, you got to know the inactive form, which is biotin, the active form, which is carboxybiotin or biocystin, C-Y-T-H-I-N. And that's what's going to perform the reactions we're going to talk about. Biotin does one thing only. It does carboxylation. Does anyone remember what does decarboxylation? No? TPP does oxidative decarboxylation, which is vitamin B1, and B6 pyridoxal phosphate does decarboxylation. Good. So carboxylation is done by B7, which is biotin, or just biotin for Dr. Sarkar's purposes. Any of your carboxylation reactions are going to be biotin. So what type of enzymes are carboxylators? Carboxylases. So if you have a carboxylase, that's going to be a carboxylase reaction. And guess what? It takes a substrate and makes it a carboxylated substrate. Real novel concept there. A couple of examples you're going to want to know, because a few of these you have seen. Pyruvate to oxaloacetate. 3 carbons, 4 carbons. And that's a big hint. If he gives you a structure and it goes from 3 carbons to 4 carbons, I don't think he gives you a lot of structures, but it goes from 3 carbons to 4 carbons, what's happening to it? It's being carboxylated. It's having a carbon added. And the main way we add carbons is through carboxylation. So in this case, pyruvate carboxylase is named carboxylase. It takes pyruvate to oxaloacetate. It's important for gluconeogenesis, and that's one of your example reactions. Acetyl-CoA carboxylase is very, very important. It is the rate-limiting enzyme in what pathway? Fatty acid synthesis. It takes acetyl-CoA to malonyl-CoA, right? Makes that important intermediate. Malonyl-CoA is the donor of all 2 carbon units. You got enough bricks? Acetyl-CoA carboxylase. It's a carboxylase enzyme. You know that. It's adding a carbon right here. It's called a carboxylase. It's important for fatty acid synthesis. Another good example of reaction that comes directly from this trimester, so he likes that one. Propionyl-CoA carboxylase. This is that intermediate step in the breakdown. In the breakdown of odd chain fatty acids. Propionyl-CoA is what you get from breaking down an odd chain fatty acid. It's turned from propionyl to methmalonyl-CoA, and then remember we talked about in B12. One of the reactions for B12 is methmalonyl-CoA to succinyl-CoA. You guys remember that? That is an example reaction you're probably going to want to know for B12. So the 2 guys that are responsible for the breakdown of odd chain fatty acids, and he's probably not going to ask you in this manner, would be biotin carboxylating it, and then B12 taking methmalonyl to succinyl-CoA. Methmalonyl to succinyl is a great example reaction for vitamin B12. This one might come up, but probably not. And he's probably just going to ask you enzyme names for this one. So what you really need to know is that carboxylation is biotin. So if you see a carboxylase enzyme, it's an example of a biotin enzyme. And last one, not really important. Methylchronotyl-CoA to methylgluconyl-CoA. I don't even know what catabolism of leucine. There you go. I was like, I don't even know what that's involved in. Catabolism of leucine. I wouldn't bother memorizing that one. Gluconeogenesis, acetyl-CoA carboxylase for fatty acid synthesis, and pyruvate carboxylase for gluconeogenesis. Those are important. But primarily look for carboxylase enzymes. That's what Biochem does. It is a carboxylator. This you do want to know. Egg whites contain a heat labile protein called avidin. It's heat labile. What's more dangerous, cooked eggs or uncooked eggs for this purpose? It's a heat labile protein, so what's more dangerous, cooked or uncooked? Uncooked. It's heat labile, which means it's fragile to heat. Labile means it can be broken down by heat. I want you guys to get that because that might come up on the test in some manner, talking about heat labile, because there's a couple of things that are heat labile. What's the one in fish that's heat labile? Thiaminase. And where would you get thiaminase in raw fish? Because it hasn't been cooked, the enzyme's heat labile. So once you cook fish, it's no big deal. Once you cook eggs, they're no big deal. Avidin is in raw eggs. It's a heat labile enzyme or protein. And it combines very tightly with biotin, which prevents absorption. So it's going to cause malabsorption of biotin. You won't have carboxylation, no gluconeogenesis, no fatty acid synthesis, no breakdown of odd-chain fatty acids, problems. Lots of random stuff on here. I wouldn't memorize them. The ones for clinical use that they talk about are alopecia and dermatitis. What is alopecia? What? Yes, specifically hair loss. So people talk about that all the time. It's one of the miracle cures for hair loss out there. I don't know if it works. I've never tried it, just if you were wondering. But lots of people do. They take biotin in the hopes that it grows hair. So, what? I'm losing my hair. I'm cool with it. I wasn't when I was younger, but now I've gotten to the point where I'm okay with it. Actually, yeah, exactly. RDA 25 to 30 milligrams. Nobody really cares. It was you guys that made the joke. No, it couldn't have been you guys because you guys didn't meet Ian, did you? A couple of tries ago, someone made a joke because Ian was tall, white guy, balding, and they're like, "What do you have to be balding and tall and white to tutor biochem?" It was a bad joke. Very bad joke. You guys are better than that. Sorry I blamed you for that horrible joke. You've heard that? How have you heard that? Oh, maybe I said it in try one. Okay. Of all the shit you remember the bad joke about balding. You don't remember all the crap in biochem. Yes, I can blame you. All right. So, any questions about biotin? Because that's it. So if I were to say niacin, you would scream back at me what? Vitamin B3. And what's niacin involved in? NAD, NADH, or I'm sorry, NAD, NADP. So what type of reactions does it participate in? What oxidation reduction. Remember, when I ask you what type of reactions, think about those, because you remember these. 6 classes of reaction from way back when. Not that this falls into the 6th class, but think about the categories of reaction, not the specific reactions, but the categories. Because NADs are in everything, right? I mean, they participate everywhere. But when he asks you what types of reactions, think about oxidation reduction, decarboxylation, oxidative decarboxylation, carboxylation, all that stuff. That's what he's going to be asking about. And there's going to be a lot of those questions on this test. The 3rd level questions might give you a reaction. They might say acetyl-CoA to malonyl-CoA. What type of reaction, or not what type of reaction is this, but what vitamin does this? Acetyl-CoA to malonyl-CoA. And you need to understand that acetyl-CoA to malonyl-CoA is a carboxylation reaction. It's going from 2 carbons to 3 carbons. It's the rate-limiting enzyme of fatty acid biosynthesis, and since it's a carboxylation reaction, it must be performed by biotin. Fantastic. It's a carboxylation reaction. It must be performed by biotin. Great job. Cool. Vitamin C. I didn't have a title on the top of page 150. I think it's on the last page. Vitamin C is a.k.a. ascorbic acid. Actually, an interesting thing, you'll talk about this in nutrition more. Ascorbic acid is the literal vitamin C compound. It's actually what vitamin C is. But when you take vitamin C in plants, it comes with a ton of bioflavonoids and other good compounds. And they've done tests on the efficacy of whole food supplementation of vitamin C versus ascorbic acid, which is what you get when you buy a vitamin C pill at like Walmart or Target or something. And the differences are phenomenal. So you may say, I'm deficient in vitamin C, I need vitamin C. And taking ascorbic acid really isn't awesome for you. It doesn't really do anything. It gives you vitamin C without all the other stuff that helps and performs and does everything. So that's a real big concept in vitamin biochemistry, is not only just the other associated flavonoids that go with all of these vitamins, but all the proteins and the fats and the enzymes and everything that comes with the fruit, which is why people always say it's best to get your vitamins from food, not from supplementation. So we're going to talk about vitamin C, aka ascorbic acid, which is the literal vitamin C compound. Now, there's this nice little pathway here. It's a 6-carbon lactone. Don't worry about it. It's synthesized from glucose. So you go through this whole pathway, and there's an enzyme in here called gluonolactone oxidase. Turns gluonolactone to ketogluonolactone. You need to know the enzyme gluonolactone oxidase, and that humans does not contain it, according to your lecture manual. Humans does not contain that enzyme. Since humans does not contain that enzyme, can we make the active form of vitamin C? No. So what do we have to do? We have to ingest it. We have to get it from plants that can, because I think it's animals can't. What? Emergency. Yeah, exactly, emergency. So once you go through gluonolactone oxidase, it can go over to ascorbic acid. Don't really need to know the product and substrate of that. You do need to know the enzyme name. Gluonolactone oxidase is very important for vitamin C biosynthesis. And since you know it's gluonolactone oxidase, you should guess that the substrate for gluonolactone oxidase is gluonolactone, because enzymes are named for the substrate and what they act on. Gluonolactone oxidase acts on gluonolactone, so that'll help you a little bit. The active form is ascorbic acid. It can be reduced to deoxyascorbic acid and oxidized to ascorbic acid. Again, not huge TQ for that one, but he did talk about it in our class, so I'm not sure if it's important for you guys. Lots of other stuff down here, food sources, citrus fruits, berries, melons, lemons. You think about all that stuff and you're like, oh yeah, that's great. That's high sources of vitamin C. One of your best sources of vitamin C? Green leafy vegetables. Eat green leafy vegetables. They're effing amazing for you. Like literally you get to clinical nutrition and the food source of every major vitamin and macronutrient and micronutrient is green leafy vegetables. So eat kale and spinach and broccoli and all that stuff. Those are, I guess broccoli's cruciferous. But okay, leafy vegetables, don't care. These are important for vitamin C. It is the least stable of all of the water-soluble vitamins. It is highly heat-labile. So what happens when you fry your bell peppers? You have no vitamin C left, but it is stable to freezing. So it is stable to freezing. We've got some nutritional counseling going on, which means that those frozen berries you get, the vitamin C is actually still good in them. The frozen vegetables, it's one of those things, and in fact most vitamins and proteins deal better with cold than they do with heat, but you know, it's better than nothing. So you need to know those three things. Vitamin C is highly heat labile. It is the least stable of all water-soluble vitamins, but not because of cold. So refrigerate your fruits. That's his clinical pearl. Refrigerate your fruits. Here's another thing. He probably won't ask about it, but just in case he does, what are the three major things that break down a vitamin? I don't know if I really hit on this. What's the one that breaks down vitamin C? Heat, water, light, and oxygen. Heat, light, and oxygen are bad for pretty much every vitamin. So where should you store your vitamins? In a dark container in the fridge. Same with your fruits. I mean, I personally cut my fruit open every day. I cut it up and bring it to school because it's a lot easier for me to eat while I'm at school having cut fruit already. But what does that mean? That means that the surface layer that's exposed to the light and the oxygen is probably not going to have as much nutritional content if I just eaten an apple as if you're supposed to. So little trade-offs. And those vitamins you get, like vitamin whatever that comes in the clear container and you leave in your cabinet above the stove, that's not great for it. You're going to lose a lot of the vitamins over time. So heat, light, and oxygen are bad for pretty much every vitamin. That's a generalization. It's, Vitamin A is used in oil form for the base. Is it the same thing as vitamin? Vitamin what? A. Vitamin A is used in oil form for the base. Is it the same thing as vitamin? Vitamin A is used in oil form for the base. Is it the same thing as vitamin? Vitamin A is used in oil form for the base. Is it the same thing as vitamin? Vitamin what? A and E are not the same vitamin. Is that what you're asking? Yes. Yes, and they're not supposed to be. Your best fish oils will come in a dark container, and your best, especially oils, are much more sensitive to light, oxygen, and heat than are water-soluble vitamins. So that's really weird, because I noticed that, because all of my old fish oils used to come in that. My vitamin E comes in a clear container, and really it's not supposed to. Now remember, it's not so much like light that comes out of a fluorescent light bulb. It's more UV light that has an effect on it. So as long as it's inside, it's better off than not. But cool. Vitamin C is absorbed as ascorbate or dihydroascorbate, which is, again, reducible, oxidizable. And this is important. This is one of those water-soluble vitamins that can be stored in the body. So normal stores of vitamin C, we do store vitamin C. What other water-soluble vitamin do we store? B12. B12 and vitamin C are the only water-soluble vitamins that we store. The rest of them we have no stores of, which means they're depleted easily. It takes 3 to 4 months to deplete vitamin C because we have 3 to 4 months of storage of vitamin C. It's about a year for vitamin B12, 3 to 4 months for vitamin C. Those are definitely TQs, water-soluble vitamins that are storable. Vitamin C is widely required for metabolism. This is a list of everything that vitamin C does. It does a tremendous amount of stuff. Electron donor for at least 8 enzymes. I would TQ and star collagen hydroxylation. It does carnitine biosynthesis. I would TQ and star norepinephrine biosynthesis. And it does amidation and tyrosine metabolism. Basically, we'll talk about that next. Down here, so the two you need to know from up here definitely are collagen and norepi. Norepi because we studied it a couple times in Chapter 8 and Chapter 9. Collagen because that's a really important one, and it makes the basis of one of our major vitamin C diseases, which I'm sure you guys know is scurvy, which is a collagen problem. It's also a really big TQ for you guys to know in clinical practice. If you see someone that's got collagen problems and capillary fragility and bruises easily, it might be something as simple as a vitamin D deficiency. And where do you get your vitamin D? Fruits and vegetables. And what do Americans not eat? Fruits and vegetables. And what do they take? A vitamin C supplement. And they're like, I get 1,000 milligrams of vitamin C. No, you get 1,000 milligrams of ascorbic acid, which does not have the same biological effect as vitamin C. So it's important that you're able to educate your patients on this stuff. And scurvy is the end game disease. That's what pirates got, and that's why they figure out they called them Lyme. We'll talk about that. Anyways, limeys. Yep. The other thing it does is any oxygenase enzyme. So collagen, norepi, and either mono or dioxygenase enzymes. What does an oxygenase enzyme do? It adds an oxygen. Really easily named. It adds an oxygen. Monooxygenase incorporate a single oxygen atom. I'm pretty sure you guys can figure that out. Di incorporate 2 oxygen atoms. Fantastic. That's what you got to know. How many oxygens does dioxygenase incorporate? Yeah. The other thing is vitamin C, TQ is an antioxidant. We talked about these guys before. ACEs. ACEs are your antioxidants, A, C, E, and SE. We've talked about C. Yeah, we haven't talked about the other ones yet. You got to know those. And remember, those go from low O2 to high O2 action. So A works best in low O2 conditions. Selenium works best in high O2 conditions. So ACEs. C is the first one we're talking about, major antioxidant capabilities. It's actually, I guess we'll get to that when we get to iron. I'll preface this now. It's really important for iron absorption. So again, clinical pearl for women who have problem with iron. And A, where do you get most of your iron? Green leafy vegetables. You can get it in meat, but ideally, the best kind of iron comes from plants because it's already in the state we want it to be. We don't have to reduce it. So we absorb ferrous iron, Fe2+, and then we oxidize it, ferric iron. So humans and animals store iron in a ferric state, Fe3+. So if you eat it, it's in a ferric state. You can't absorb ferric iron. You have to turn it into ferrous iron. Vitamin C turns it into ferrous iron. So if you're not getting vitamin C with your meat, you're not absorbing the iron from your meat anyway. So you need to make sure you're getting all this stuff, or they're eating green leafy vegetables, which has the normal amount of iron in it anyway. So Intracellular functions, it does electron donation, DNA repair, DNA transcription, mRNA, protein stability, apoptosis. Here we go. It scavenges free radicals and protects against oxidants and other oxidant-mediated damage. It's an antioxidant. Now, I want you to put that as a TQ. It's extracellular antioxidant. It is an extracellular antioxidant. Once in a blue moon, he'll ask which ones are intracellular, which ones are extracellular. We'll talk about, I think he has one of the intracellular ones later. But vitamin C is an extracellular antioxidant that works primarily at a lower O2 concentration. Lipid peroxidation, LDL oxidation, those are antioxidant capabilities. But extracellular. It also regenerates the lipid-soluble vitamin E from its oxidized form, which means that one of the primary purposes of vitamin C as an antioxidant is to keep vitamin E, which is an antioxidant, unoxidized. So vitamin E will go and reduce something, it will antioxidize something, and then vitamin E is spent. So vitamin C comes along and antioxidizes it, which is reducing it. So vitamin E is then free to go and antioxidize something else. So vitamin C will continually help these other antioxidants to stay reduced so that they can continue to be antioxidants. Does that make sense? Okay. Prevents atherosclerosis. That's fantastic. Not probably going to test you on that little point. And here we go, TQ, TQ, TQ. It increases the absorption of iron in the small intestines because it keeps iron in the reduced form. Vitamin C increases the absorption of iron. You're going to get hit with that all the way up through lab diagnosis. Like pretty much once per trimester, you might skip try 2 with that one. You'll get at, oh, you're in try 2. Might skip try 3. I don't know if physio hits you on it. But it's very important. You're going to get it in clinical nutrition. You're going to get it in the lab diagnosis. That will keep coming up. Vitamin C reduces the following compounds: oxygen, nitrates, cytochrome. Cytochromes, don't worry about it. Reduces is antioxidizes, right? Because an antioxidant is something that reduces. It does the opposite of oxidation. So AKA for, and don't call it this, nobody's going to take you, but an antioxidant could also be called a reducer. Now, you're not going to forget this one, I know, because this one's really easy. Vitamin C deficiency is scurvy. Primary signs: bleeding gums and hyperkeratosis, increased skin cells around follicles. Bleeding gums and hyperkeratosis. This is a pirate disease. The reason they figured it out is because pirates used to take stuff like salted meat and anything else that could be stored on a ship to sea. They didn't have any fruits or vegetables or green leafies because those things didn't travel well in a boat. And these guys were coming back and they were getting bleeding gums and, they were falling apart because they couldn't make collagen and they couldn't repair collagen. So everything that required collagen, which is all of your tissues, would just go to shit. So easy bruising, capillaries exploding, hemorrhaging, and one of the primary signs is bleeding gums. And what these guys finally figured out somehow is some batch of pirates ate a bunch of limes one time, or landed on an island and ate limes, and they didn't get scurvy. So from then on out, they're like, oh, we're going to send all the pirates with limes because limes prevent scurvy, which is why pirates were called limeys. Hey, guess what? Limes have enough vitamin C, and they travel really well. Limes don't go bad forever. You can keep them in your house for drinks whenever you need them. So they called them limeys for a long time until they figured out that it was actually the vitamin C in the limes that was the good part. Cool? Limeys, scurvy, pirates, it's all good stuff. Scurvy. And yeah, you got to say it like that. I'm sorry, scurvy. Why can we get a vitamin C excess? What's key about this water-soluble vitamin that allows us to get an excess? It can be stored. Can't get excesses of B1 through B12. I'm sorry, B1 through B11. Not that B11 exists. Unless you take inane doses of them. But B12 and vitamin C, you can get toxicity or excess. Toxicity of vitamin C greater than 1,000 milligrams a day can form oxalate, which leads to kidney stones. It's a stone former. It leads to kidney stones just like calcium excess will, supposedly. I've never seen this, and I know lots of people that take 2,000 milligrams a day and don't have kidney stones, but I'm not going to argue with Dr. Sarkar. This is the one RDA you got to know. I'm going to tell you this right now. Don't write it down yet. The RDA is 70 milligrams a day, according to whomever. The TQ for vitamin C is 100 milligrams a day. TQ, vitamin C, RDA, recommended daily allowance, 100 milligrams a day. Guess what? You can also write 0.1 grams. 0.1 grams and 100 milligrams mean the same thing. Got to know your metric conversions. So he will probably write 100 milligrams, but be prepared for 0.1 grams. I think that's as far as he'd go. He won't stretch it any farther than that. Like what is it, 100? So what would that be? Like 1 million nanograms or something like that? Yeah, don't worry about it. 100 milligrams a day is the TQ for vitamin C RDA. It's the only RDA you need to know in water-soluble vitamins, at least according to what he taught us a couple tries ago. So if he told you otherwise, please say so. But when he talked to us, he made it very clear that you don't need to know any RDAs except for vitamin C, TQ, 100 milligrams a day. That's chapter 11. Questions? No, you don't get to go home yet, because I know someone was going to raise their hand and ask. Okay, we got to get to fat-soluble vitamins, and then you guys can get out of here. I'm not going to go any further. To my knowledge, your test is, I have no idea. Oh, holy crap. Where is he lecturing right now? Sweet. Traditionally, your test goes through fat-soluble vitamins, through lecture 12. That's what it's supposed to do. It's supposed to go 9 through 12. Was 9 on this test? I think so. 9's all the special products. 10 is metabolism. 11 is water-soluble vitamins. 12 is fat-soluble vitamins. That's traditionally where it is, and then he leaves macro and micronutrients for the final. So we're going to get through fat-soluble vitamins tonight, and then if we need to, we may do water-soluble, or we may start the other stuff next week. The next chapters are really easy. It's, hey, you need to know this, hey, you need to know this. But it's kind of like this where you just got to memorize it. Please know your vitamin chart. The vitamin chart has both fat-soluble and water-soluble vitamins. It's on the web page that you guys hopefully have a link to by now. So please know that stuff, because that will definitely be TQs on this test. In the metabolism chapter, know the basics. Oh! Shoot, this is a question I missed. I just remembered it from the metabolism chapter. I think it's the greatest storage of energy in the body. Where is the greatest storage of energy in the body? Back from the metabolism chapter. And he said glycogen in muscle, glycogen in liver, triglycerides in adipose tissue, or something else. It's triglycerides in adipose tissue. I thought it was glycogen in liver, because I was like, some people aren't that fat. And he's like, it doesn't matter. He's like, no matter how thin you are, he's like, the most energy storage you have as a human is triglycerides in fat, or in adipose tissue. Glycogen in the liver provides a ton of energy, but it can't compete with body-wide storage of fat. So please know that metabolism, best energy storage for your body is triglycerides in the liver. Most other people got it off common sense, but I was like, I know my biochem. I'm going to logically think this through and miss this question. You have no idea how many times I do that. I'll miss dumb questions because I'm like, well, this, and they're like, you're thinking too much about it. Just answer the simple one. Especially with Adwani. Watch out for her. All right, so fat-soluble vitamins. We did water-soluble vitamins, which are your B vitamins, and biotin and folate. Fat-soluble vitamins are A, D, E, and K. You can memorize CADE, K-A-D-E, or you can memorize ADEC, whatever you want, or you can just know it's A, D, E, and K. Fat-soluble vitamins. Couple of key points we're going to go over before we get into each of the individual fat-soluble vitamins, and we do go into more detail on most of these guys than the water-soluble vitamins. So there's a few big hitters in here that we're going to spend a little bit of time on. They are apolar hydrophobic molecules. What does that mean? What's apolar? Not polar, not necessarily charged, but they don't have a pole. But you're right, usually it has to do with charge, which means they don't have any electronegativity, any charged groups. So they're apolar. They have no polarity. And hydrophobic means they hate water. Good. So apolar hydrophobic molecules, because they're fat-soluble vitamins. If they're fat-soluble, they're hydrophobic or lipophilic. Big concept, easy. All fat-soluble vitamins, TQ, CQ, are derivatives of isoprene units. Where did we see isoprene units? A hairless, manly Indian. Cholesterol synthesis. We made isoprenes. So in cholesterol synthesis, you take a bunch of acetyl-CoAs, you put them together, you get an isoprene, you take some isoprenes, you put them together, and you get cholesterol. Fat-soluble vitamin synthesis, you take a bunch of acetyl-CoAs, you put them together, you get isoprenes, you turn them into fat-soluble vitamins. Not all fat-soluble vitamins are derivatives of cholesterol, but they are derivatives of isoprene units, which are made from acetyl-CoA, which are fat-building blocks. So please know they're all isoprene subunits that usually comes up on one of your on your test. They're derivatives of isoprene units. They cannot be synthesized in the body in adequate amounts, which means their source must be dietary. And this is huge. Absorption is regulated by the GI tract in the same way as dietary fat. Absorption is regulated in the GI tract in the same way as dietary fat. That means that if you cannot absorb fat, you cannot absorb fat-soluble vitamins. Stuff like bile salts, your gallbladder, cholecystitis, which is gallbladder dysfunction. All of that stuff that allows the formation of my cells and the emulsification of fat and the breakdown of fat into small pieces so it can be broken apart and absorbed. If you don't have that intact, you do not take in fat-soluble vitamins nearly as well as you should.
This is the security department. Please be advised that the bridge gate and creek lot will close at 6 p.m. Please move your vehicle from the creek lot to the main campus at this time. Thank you.
I'm so sorry. I don't get a red light passes, I swear to God. Be like, haha, walking give me those tickets.
It's not him you got to worry about. He'll walk. It's the other guys that won't. So fat-soluble vitamins need fat absorption intact. If you have some problem in your intestines that you're not absorbing fat, it could be lymphatics, it could be gallbladder, it could be the enzymes, the lipases that break down fat, the bile acids that are cholesterol-based, all of that stuff. So if he talks to you about some sort of fat malabsorption, the first thing you're going to look for is a lack of a deficiency of a fatty acid. Fat malabsorption leads to deficiencies. Not fatty acids. Fat malabsorption leads to deficiencies of fat-soluble vitamins because you can't absorb them. And then the nice thing is they're fat-soluble vitamins. So can we store them? Yes, we can store them. We can store fat-soluble vitamins easily. So we don't usually get deficiency conditions unless long-term chronic malabsorption or dietary insufficiency. What can we get with fat-soluble vitamins that were harder to get with water-soluble vitamins because we can store fat-soluble vitamins? We can get toxicity conditions. So pretty much all of our fat-soluble vitamins, we're going to talk about toxicity. We're also going to talk about some deficiency conditions, but deficiencies are a little harder to get as long as you have proper dietary intake and you can absorb them. But if you go deficient for, a month or two, no big deal because you have a storage. Where do you think these guys are stored? Fat, adipose tissue. Yep. So, requires normal fat absorption, absorbed along with dietary fat. Bile salts, lipoprotein lipase are all required. TQ, any condition or diseases of the biliary system, that's gallbladder or liver. And anything that interferes with fat absorption can lead to malabsorption of fat-soluble vitamins. So just like we've been talking about, if you can't absorb fat, you can't absorb fat-soluble vitamins. It's like the 5th week in a row I've broken that. Just brushing it. Not even fair. They are transported to the liver bound to chylomicrons. Ooh, does that sound familiar? Probably not a TQ for this chapter, but that's how we transport fats. That's how we transport fat-soluble vitamins. They're transported by lipoproteins in the blood because they are not soluble. Fat-soluble vitamins are hydrophobic or lipophilic. They are not water-soluble. They must be transported. And they are stored for varying amounts of time. Oh, here you go. Vitamins A, D, and K are stored in the liver primarily, and vitamin E is stored in adipose tissue. May want to TQ that. Vitamins A, D, and K are stored in the liver, and vitamin E is stored in adipose tissue, which is really lucky because E is an antioxidant, and one of the most dangerous things you can have is oxidized fat. So it causes atherosclerosis. So having vitamin E stored in your fat is great because it keeps all your fat reduced, non-oxidized, antioxidized. Cool. Excretion. They're not excreted in the urine because they're not water-soluble. They're more likely to appear in the bile and thus excreted in the feces, etc., etc. Toxicity occurs for at least vitamins A and D overdosage. We'll talk about those when we get there. Here's your biomedical importance. Here's a nice little refresher. Refresher. Once you study this chapter, once you come back and it's the night before the test, you're like, crap, which deficiency conditions were associated with each of these? Vitamin A deficiency leads to dry eyes, xerophthalmia, and night blindness. Cool. What's Accutane do? Maybe. That might be more associated with the fact that it's a drying agent itself.
I mean, how, can I ask you a question?
Mm-hmm.
I was just curious about this. When you take, it's a retin-A, so you can overdose on retin-A, right? And if you
Yeah.
have a high level that in your body, is that why, it was a cancer, they were giving it to female, and they ended up giving it back and she died out years later from it. So.
But is retin A a vitamin A derivative?
Yeah.
Because that would be the opposite of what we're talking about.
I know, but it still has the same effect, that's why I'm like,
I have no idea. I'm not real familiar with the mechanism of action of retin A, but I mean vitamin A deficiency causes dry eyes and dry skin and stuff like that. Not so much an excess of. I'm not sure if it's some sort of weird derivative or if it soaks up vitamin A in your skin to use it as as part of the medicative process and it depletes the rest of vitamin A. That might be something, or what it does. But I mean, most acne treating drugs are drying agents in themselves, so they tend to cause dry skin, not necessarily so much due to the vitamin A, but just because they figure if they dry out the environment, the propionic bacteria that cause acne can't live. You guys know that now, right? That's part of propionic vulgaris. Can't live, so they dry it out. So that might be more the cause of dry skin. I don't know if it's vitamin A. No? That's cool. That's stuff I need. We're starting to get into the drugs as we go along. Farm Tox is in try 5 where I am. So we're starting to get some of the drugs under our belts. But stuff like retin A and stuff like that I have not encountered at all. So vitamin D, rickets and osteomalacia. Rickets is osteomalacia in kids. It's soft bones. Vitamin D is really essential for one thing, and we'll talk about it. It's calcium absorption. Very essential for calcium absorption. If you don't have calcium absorption, you don't have vitamin D, you get soft bones. And bowed legs, yep, from non-weight bearing. Like if your bones can't hold up to the stress, they bow because curves hold stress better. Vitamin E, neurologic and anemia. Anemia, really? I do not remember that at all, to be quite honest. And vitamin K, big, important for blood clotting. Vitamin K will be important in bleeding and hemorrhage. We're going to talk extensively about that. You will talk about that a lot. Okay. Vitamin A. Leading cause of vitamin A deficiency is the leading cause of blindness in the world. Vitamin A deficiency is the leading cause of blindness in the world. Vitamin A deficiency is the leading cause of blindness. I had it right the first time. Dang it. Leading cause of blindness in the world. You guys get up here and talk. Lack of vitamin A causes dry eyes, etc. But I would know the leading cause of blindness in the world is vitamin A. I'm just going to roll with it. Did you really? That's awesome. Just like the previous chapters, you need to know the active form, the inactive form, the reactions or what it does, and deficiency and toxicity conditions, just like previous chapter. So biologically active forms of vitamin A are classified as retinoids. Vitamin A is a there's different retinoids, but vitamin A is active forms are retinoids. So vitamin A is the inactive form. It's got 3 active forms: retinol, OL, retinal, AL, as in alcohol aldehyde, and retinoic acid, as in carboxylic acid. So those are the 3 forms you can oxidize and reduce back and forth, right? We talked about that. You can take an alcohol and you can oxidize it to an aldehyde. You can take an aldehyde and you can oxidize it to a carboxylic acid, and you can reduce them backwards. So these are 3 interconvertible active forms of vitamin A. Now I would know right here, retinol, OL, is full vitamin A activity. The rest say some, which means he might ask you what is the most active form of vitamin A, and you need to know the most active form of vitamin A is retinol, alcohol, OL. Alcohol is good, it's the most active, that's when you're jumping around and all that stuff. Might help, I don't know. Retinol is the most active form of vitamin A. Now, they're each essential for different components, so don't get me wrong when I say that would just be a straightforward question. What's the most active form of vitamin A, or what's the most biologically active, or something like that, form of vitamin A? That's retinol. But each of those is going to have its own components, and if he asks you about each of those components, you need to pull up the right one. In animal fats, it is stored as a retinol ester. I wrote that in. I don't know if it's in your book. You might want to write it in. In animal fats, we get it as a retinol ester, R-E-T-I-N-Y-L ester. What is that a storage form of? cholesterol esters, storage form of cholesterol in animals, right? So what do these guys do? They take a cholesterol and they esterify a retinol to it. And when you get a retinol ester, that's how animals store it. So it's the storage form of vitamin A in animals. In vegetables, it exists as beta-carotene, which is a yellow pigment. Beta-carotene, which is a yellow pigment. I don't know if the yellow is too important. He probably won't ask you that. But you do need to know beta-carotene is in plants and retinol esters is in animals. Well, there's two types of vitamin A storage we can get. Vitamin A is destroyed by sunlight, just like pretty much everything else, especially the fat-soluble vitamins. They're very easily oxidized by sunlight, by heat, by oxygen. We get it in a lot of different stuff, especially fatty stuff. So fish oils becomes fortified in milk now. Eggs, butter fortified with it. Saltwater fish, marine mammals. You get it in dark green and orange fruit. Now, please notice, beta-carotene is a yellow pigment. That does not mean that the fruit is going to be yellow. Yes, yellow peppers are probably very high in vitamin C, but one of your best classic sources of vitamin C is, one of your best classic sources of vitamin A for beta-carotene is carrots. Everyone told you eat carrots, you can see in the dark. Carrots are orange, but it's a combination of all the vitamins and minerals in them that gives them their true pigment. So green and orange fruits and vegetables, carrots, spinach, sweet potatoes, broccoli, all that stuff, great sources of vitamin A. And you don't think about vegetables having a lot of fat in them, but they do have fat-soluble vitamins in them, which are really good for you. So that's where most of the fat comes from. Source of color in produce, but can be masked by chlorophyll. That's your explanation for why yellow pigmented vitamins that are sources don't always end up as yellow fruits and vegetables, because chlorophyll is there, and chlorophyll is primarily what color? Green. Good. Chlorophyll is green. Okay. So we can interconvert it. Please note that once it goes to retinoic acid, it cannot go backwards. That might be a TQ. Once it goes to retinoic acid, that is a final conversion. It cannot go backwards. Retinol and retinal are interconverted in the presence of NAD or NADP-dependent dehydrogenases or reductases. So what vitamin do we need to interconvert them? What? Niacin, a.k.a. vitamin B3. Okay, just throw some random numbers out there, aka vitamin B3. So you need vitamin B3, aka niacin, to convert these guys because it's NAD, NADPH dependent, because it's an oxidation reduction reaction. So going from a primary alcohol to an aldehyde to an ic acid is an oxidation reduction reaction. Once formed from retinol, TQ, retinoic acid cannot be converted back. So once formed, retinoic acid is final. Retinol esters are dissolved in animal fats. They're dispersed in bile to produce micelles, hydrolyzed in the intestines, absorbed. We get free retinol, it's absorbed. It goes to chylomicrons, it goes to the liver, et cetera, et cetera. Normal fat absorption, just like everything else. Now, beta-carotene, you need to know. Beta-carotenes are present in plants. Ingested beta-carotenes are oxidatively cleaved by beta-carotene dioxygenase. How many oxygens does that add? Two. And what vitamin does that? Yeah, nice job. Vitamin C. Remember, the oxygenase enzymes, monooxygenase, dioxygenase, as well as collagen hydroxylation and norepinephrine biosynthesis, are vitamin C dependent. So beta-carotenes present in plants are cleaved by a vitamin C dependent enzyme called beta-carotene dioxygenase. I would know that. Beta-carotene dioxygenase, which forms retinaldehyde. Retinaldehyde reductase will take retinal to retinol, because going from an aldehyde to an alcohol is reducing it. Going from an aldehyde to an alcohol is reducing it. You don't need to know that, but you can infer that from the enzyme. And if you remember that alcohol is oxidized to aldehyde, is oxidized to carboxylic acid, or they can be reduced backwards, it makes these enzymes a little easier. Retinal can be reduced to retinol in the presence of the enzyme retinaldehyde reductase, which is NADPH dependent, because most biosynthetic reductive processes use NADPH. So I would know those two enzymes. Those are important for the formation of vitamin A from plant sources. Animal sources, it comes preformed. It comes ready to go. We just got to break it down and absorb it. Plant sources, we have to do some conversions. Please note that it uses NADPH. He did make emphasis of that when we went through this program. But remember, biosynthetic reductive reaction probably uses NADPH. All right, small fraction is oxidized. Most of it's esterified and stored as a retinol ester because animals store it. Here you go. Retinol is esterified with a saturated fatty acid to make a retinol ester because that's how we store this stuff. And it's incorporated into lymph chylomicrons and spread out through the blood where it is stored in the liver primarily or delivered to the tissues. Nice little flow chart of how everything's going on. I'll run through it. Not a lot of TQs come through here. In the lumen, we can get a retinol ester from animal fats, broken down to retinol, absorbed, turned into a retinol ester, put into chylomicrons, and then it can be stored in the liver as a retinol binding protein bound to retinol, or it can be shipped out into the blood as retinol esters. Beta-carotenes do the same thing, beta-carotene dioxygenase, and then that'll turn it into retinol, Beta-carotene dioxygenase will turn it into retinal, and then retinaldehyde reductase will turn it into retinol, and then same pathway. So, overview of what we've been talking about. You can add retinal right there and the two enzymes for beta-carotene if you care, but this is what we've been talking about. Other than that, not a lot of TQs. Now here's the fun part. It's stored as retinol ester. Don't worry about that. He may ask you stuff about this. There are three ways it's transported. Retinoic acid binds to albumin. Where would we bind it to albumin? In the blood. Retinol binds to apo-retinol binding protein in liver cells. Or retinol binds to cellular retinol binding protein in other tissues. So if it's in the liver, it binds to apo-retinol binding protein. If it's in another tissue, it binds to cellular retinol binding protein. If you see a binding protein, what does it do? It binds that guy. Retinol binding protein, thyroid binding globulin, et cetera, et cetera, et cetera. There's a ton of those guys. Doesn't ask a lot of those, but just the general overview. If you see this TQ, if it's in the liver, it's an apo. If it's not in the liver, it's a cellular. And that's for retinol, which is the most active form, OL. Now, here's where the TQs come from, or start to come from. Retinol, OL, supports reproduction, and it supports gene expression, and it's important for activation and proliferation of lymphocytes, which means it increases immune function. So vitamin A is important for reproduction, gene expression, and immune function. Retinol, alcohol, vitamin A is important for reproduction, gene expression, and activation. Now, he did mention this, and we'll talk about this a little later. This specifically is very contraindicated in pregnancy because it can build up to a toxic level real quick in a baby. It can build up to a toxic level very quickly in a baby because they're such a small organism and it goes through the blood. So vitamin A, especially retinol, is very contraindicated in pregnancy. Important for gene expression, reproduction, and lymphocytes or immune function. Retinaldehyde, AL, is important for night and color vision in the retina. It's important for vision. Retinal is important for vision. We're going to talk about rhodopsin. It's present for the rod cells, et cetera, et cetera. In fact, I think we're going to talk about that right now. Okay, so he goes into a lot more detail than we had in our lecture manual, so I have some stuff written in. We're going to go through the basic part first, then we're going to go through the complicated part, and I hope when he covers this in your class, it makes a hell of a lot more sense because it took me weeks to get this down. In fact, it took me like 2 trimesters to get this down because I missed it on the test. So, retinal is important for night vision, which means it's present in rods because rods are the visual receptors in the back of the eye in the retina that pick up light light stimuli. Cones are important for color, CNC. Cones are important for color. They're better detail vision. Rods are just general light and dark. So at nighttime, that's why you don't see colors real well because there's not a lot of light around. So your cones aren't very active. You don't see colors. But you do pick up a little bit of just vision in general, which is more the function of your rods. So these guys are present in rods, which is why we get rhodopsin is the visual pigment. So we get the first step here is we get all trans retinol. All that means is all of its double bonds are in the trans position. We convert all trans retinol to 11-cis retinol by the enzyme retinal isomerase. I'm sorry if I'm saying retinol, it's supposed to be retinal. So all trans retinal aldehyde is converted to 11-cis retinal by retinal isomerase. Cool? Then 11-cis retinal combines with opsin to to form rhodopsin. 11-cis-retinol combines with opsin to form rhodopsin. Rhodopsin is present in the rods. All of this happens in the rods. On exposure to light stimulus, we get a calcium influx that cleaves rhodopsin, and we get all trans-retinol and opsin again. So it's a cyclical process where we keep having this happen. Now, that we're going to go more into depth on what happens in that. But we get the formation of rhodopsin and then the breaking of rhodopsin due to light stimulus. So just in the dark, you form rhodopsin. In the light, you break rhodopsin. Very basic. Make sure you know that much. He might ask you a test question on that. In the light, you break rhodopsin. In the dark, you form rhodopsin. When rhodopsin is exposed to light, it's dissociated and bleached and forms all trans-retinol and opsin. Calcium channel in the membrane of the rod cell in a GTP-dependent manner. That's what the calcium influx comes from. Calcium influx increases, which triggers a nerve impulse, allowing light to be perceived by the brain. So the process gets a calcium influx, the breaking of rhodopsin, and a stimulus to go to the brain because the cell depolarizes. Or at least I think those guys are not action potentials, but they get summated. So the cell depolarizes a little, it sends a stimulus to the brain, you get the perceiving of light. So that's what that is responsible for. Now we need to go a lot more into detail because I think that's all he had in our lecture manual. So, where are we going to start? There's a protein you need to know called transducin. A protein you need to know called transducin, which is activated by light. Transducin is activated by light. Now, to my knowledge, transducin is a G protein. Transducin is a G protein. When it is activated by light, it activates cyclic GMP-dependent phosphodiesterase. When it's activated by light, it activates cyclic GMP-dependent phosphodiesterase. Is this right in the light? Yes. Okay. So up here I've got this premise written down. So in the dark you can't see. That's what we're going to start with. In the dark you can't see. Your levels of cyclic GMP are high in those conditions. Your levels of cyclic GMP are high, and I would write this down because these are where the T2s come from in this area. In the dark you can't see. Your levels of cyclic GMP are high. You have a light stimulus that causes depolarization of the cell. Light stimulus causes depolarization of the cell and consequently breaks rhodopsin. Breaks rhodopsin. You're going to get a sodium influx. You're also primarily, I'm going to change that to calcium because that's more important. You're going to get a calcium influx, which is more of what you need to focus on here. And T-alpha is activated. T-alpha is transducin alpha. Transducin is a G protein, so G protein. Write that down there. Transducin is a G protein, and when the light hits it, it's activated. The alpha subunit is activated. What does the alpha subunit of a G protein coupled receptor do? It picks up a GCP and it floats over to adenylate cyclase, or in this case, guanylate. Oh, actually, I'm sorry, that's a different pathway. Never mind. It's a different G protein. So when T-alpha subunit is activated, which means it's bound to GTP, because all G proteins are active when they're bound to a GTP. I don't know if he'll ask that, but that's straight out of last try. So when T-alpha is activated, it activates cyclic GMP phosphodiesterase. It activates cyclic GMP phosphodiesterase, which is PDE. Cyclic GMP phosphodiesterase. When cyclic GMP phosphodiesterase is active, what does it do to the intracellular levels of cyclic GMP? What does the phosphodiesterase do? Break it or make it? Breaks it. Phosphodiesterases break cyclic GMP. So T-alpha is transducin. It's a G protein. In the presence of light, it activates cyclic GMP phosphodiesterase, which decreases cyclic GMP, which allows you to see in the light. Under those conditions, the cells are hyperpolarized. Under those conditions, the cells are hyperpolarized. So, it's the dark, you can't see, you get some sort of your cGMP levels are high, you get some sort of a light stimulus, it activates transducin, which is a G protein, which activates cyclic GMP phosphodiesterase, which breaks cyclic GMP, which decreases the levels of cyclic GMP, which hyperpolarizes the cell, which allows you to see in the light. Because what does the rod do when it gets a light stimulus? What happens to it? Does it fire, right? So rods are receptors for light. So in the dark, if you get a light stimulus, if I shine a flashlight at you, your cells fire, your rods fire, they stimulate your brain that there's been a stimulus. So what happens if it's sunlight outside? Your cells need to be hyperpolarized, which is way down. Your cells need to be hyperpolarized, not depolarized, so they don't fire to your brain. Because otherwise the rods are going to be firing all the time and overstimulating your brain, saying there's light everywhere and you won't actually see anything. Does that make sense? So the stuff I missed on this chapter was what were the levels of cGMP in the dark and the light, and what were the polarization states of the rod in the light and the dark. So in the dark, your cGMP levels are high, and your cells are, in the dark your cGMP levels are high and your cells are normal or they're depolarized. In the light, your cGMP levels are low because of transducin, which is a G protein which activates cGMP phosphodiesterase, and your cells are hyperpolarized. And it's really easy, I think I just figured this out, it's really easy to remember why they're hyperpolarized, because it's light, and these guys perceive light. You have to hyperpolarize them, otherwise they'll just keep firing, and all you're going to see is white out light in a sunlit or a bright room. So, you need to know transducin is responsible for this. You need to know that in dark conditions, you have high cGMP, and you have, he might say depolarized, or he might say just normal cells. But in the light, you have low cGMP, and you have hyperpolarized cells. And that's because of the action of transducin. Does that make sense to you guys? Kind of? Okay. TQs for this section. Rhodopsin is cleaved in the presence of light. It normally is rhodopsin is retinal, retinaldehyde plus opsin. Transducin makes cGMP phosphodiesterase. Transducin is activated when there's light. It's a G protein. So in light conditions, you have low cGMP and hyperpolarized cells. In dark conditions, you have high cGMP and normal or depolarized cells. That's where your test questions are going to come from for retinal. And he's going to spend a little bit of time on it in class. So now that you know it the first time, because like I said, that took me like 4 trimesters or 2 trimesters to figure out, you're going to go to Dr. Sarkar's class and you're going to poke each other when you get to that point, and whoever's listening on vodcast, and you're going to say, hey, wake up, pay attention. This is the part that Dr. Sarkar loves to talk about, and maybe it'll make a little more sense now that Paul's talked me through it once. So make sure you pay attention to that section, especially because you might get a couple TQs on there, and I want you to understand that process. Okay, that was retinal. So retinol, OL, is important for reproduction, gene expression, immune function. Retinal is important for night vision. Retinoic acid is important for cellular differentiation, lubricants, and antioxidant capacity. Retinoic acid is important for cellular differentiation, lubricants, and antioxidant capacity. And you can also, if you care for your own purposes on here, he doesn't usually ask about it, but differentiation and maintenance of epithelial cells. So, retinoic acid is the one that keeps your skin, your mucus surfaces, all of that stuff healthy. It's responsible for mucus production, so for the eye, which is why it prevents the dry eyes, for the skin, which prevents hyperkeratosis, which is going to be some of our deficiency states. So, make sure you know that oic acid is cellular differentiation, lubricants, and antioxidants. Now, it's big, bold, underlined here, not bold. Low oxygen concentration, which is why I told you that ACEs is your antioxidants, and vitamin A is low oxygen, selenium is high oxygen concentration. It's just the preferences they have, and that's important for this chapter, that vitamin A is effective at low oxygen concentration. Now, we've talked about most of these guys. Because vitamin A does all that stuff, vitamin A deficiency causes night blindness. Because if you have a vitamin A deficiency, you can't break, you don't have rhodopsin, you can't break rhodopsin, you don't get a calcium influx, you don't get depolarization of the cells, you don't get a stimulus to the brain. I'll say it again. Because you don't have vitamin A, you don't have retinal, you don't make 11-cis-retinal, it doesn't combine with opsin to form rhodopsin, you don't break rhodopsin to get a calcium influx to get a stimulus to the brain to perceive light, you are blind at night. Cool? So, night vision, night blindness, it causes dry eyes, which is more retinoic acid. And as you can see here, if you want to, I wrote in the suffixes for which ones are important. So, night vision, night blindness is retinal. Xerophthalmia is retinoic acid. That says blindness. That is wrong. Xerophthalmia is not blindness. Xerophthalmia is dry eyes. Xerophthalmia is dry eyes. That's retinoic acid. Keratinization of epithelial tissues, eye, lung, GI tracts, that's oic acid. It's important for epithelium and lubricants. Reduced mucus, oic acid. Immune depression, retinol. Failure of cellular differentiation, because cellular differentiation is oic acid, at the very top of this page. Failure of cellular differentiation is oic acid. Reproductive abilities is retinol. What's the most active form of vitamin A? Retinol. Good. Now, that's where the other TQs come from this chapter are which part of vitamin A or which retinoid, or he may not even ask you that. You just say, he probably will. Which retinoid is important for cellular differentiation? Which retinoid is important for immune function? Which retinoid is important for night vision? And you need to know which of the three, retinal, retinoic acid. But the most active version is retinol. I don't know why it has the shortest section in here, but that's what he says. Okay, those are deficiency conditions. Now, we talked about some toxicity conditions, vitamins A and D especially. Vitamin A toxicity, greater than 15,000 milligrams a day. That's quite a lot of vitamin A. Cellular disruption and death, yada, 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 toxic effects. Yeah, probably don't worry about it. Yeah. Probably not going to ask you about that. But you can get toxicity conditions. Please memorize you can get toxicity conditions of especially vitamins A and D. He doesn't talk about any specific on here, though. I think he tells you to memorize all the RDAs for the fat-soluble vitamins, all 4 of the RDAs. So please make sure you know this. It's 1,000 milligrams a day for men and 1,000 retinol equivalents for men and 1,000 retinol equivalents for women, and 1 retinol equivalent is a microgram. So it's equivalent to 1,000 micrograms for men and 800 micrograms for women. You can ask him that in class if he wants you to memorize those. I would for both these chapters. Talk to the water-soluble vitamins, fat-soluble vitamins. Ask him which RDAs you want to know, because he will flat out narrow it down for you. I think he said no the fat-soluble and only no vitamin C for us. He did. So I'm telling you that now. Questions on vitamin A? I know that's kind of a wicked one with the retinal. Cool. Quiet. 3 more. Excited? That was a long one. I think that was the worst one. I don't remember. Vitamin D is classified, TQ, as a steroid prohormone. Vitamin D is actually kind of a steroid. They just classified as a fat-soluble vitamin, but it is classified as a steroid prohormone because it shares a lot more similarities with steroids than pretty much anything else. It is created by sunlight on the skin. We're going to talk about that. Vitamin D synthesis occurs when you get exposed to the sun. So where might you see vitamin D deficiencies? People that live in bubbles, as long as they don't take their bubbles outside. The elderly, people that live in northern climates or, very northern climates, so anywhere it's dark, especially those places where you get really far north and you get long periods of nighttime. People where it's cold, where they go outside and they're bundled up all the time. The elderly is one of the big problems, just anywhere, because they don't tend to go outside much. But you really don't need much. You need, I think it's like 20 minutes a day of normal sun exposure synthesizes 20,000 milligrams of vitamin D. So you get a massive dose from being outside, even for a little bit. So you don't need a lot, but a lot of people don't get it. So synthesized by sunlight on the skin, and light-skinned people, 15 minutes of exposure to the sun daily is sufficient to produce vitamin D. There you go. Elderly have diminished conversion ability, and they tend to spend their days indoors. Vitamin D has a key role. Please TQ this in calcium and phosphate. I'm going to drill this into your head by the end of the chapter. And guess what? You've got a section in Physio 2 on vitamin D, so if you pay attention now, it'll be good. So, heating and general food processing does not destroy vitamin D. One of the rare exceptions probably won't be a TQ, but it is safe to heat and food process it. You got to know these two. Vitamin D has two precursors. In plants, the precursor is ergosterol. Ergosterol. In humans, it's 7-dehydrocholesterol. 7-dehydrocholesterol. 7-dehydrocholesterol is present in the skin of humans. You open up the light. We're going to talk about this on the next page, but you can highlight this right here. No, you don't worry about it. Okay, here's what we're going to be looking at. Do you guys have this chart somewhere? This is great for vitamin D synthesis. This is what you want to come back and memorize. So, we're going to be talking about vitamin D synthesis. Precursor, 7-dehydrocholesterol. 7-dehydrocholesterol is present in the skin of animals. It doesn't say that. Oh, yeah, it does. It doesn't say that on, or it says that on there. It's cleaved to cholecalciferol. It's cleaved to cholecalciferol by the presence of photolysis from UV radiation. So, 7-dehydrocholesterol is cleaved to cholecalciferol by light. This metabolism occurs in a couple of different tissues, so make sure you know that. Cholecalciferol is taken to 25-hydroxycholecalciferol by 25-hydroxylase. By 25-hydroxylase. Now we've got two paths from there. Oh, and I'm sorry, 25-hydroxylase is present at the liver. So the skin is important, the liver is important, and then 25-hydroxycholecalciferol is taken by the kidney enzyme 1-alpha-hydroxylase, which is the rate-limiting enzyme of vitamin D synthesis, to 1,25-dihydroxycholecalciferol. So, you get a cholesterol precursor, your steroid prohormone vitamin D, which is 7-dehydrocholesterol. It's cleaved by light to cholecalciferol on the skin. It's added, it's hydroxylated at the 25 position in the liver to 25-hydroxycholecalciferol. Easy enzyme, 25-hydroxylase makes it 25-hydroxycholecalciferol. And then the rate-limiting enzyme for vitamin D biosynthesis, TQ, is 1-alpha-hydroxylase. 1-alpha-hydroxylase is present in the kidney, and it takes it to 1,25-dihydroxycholecalciferol, which is a.k.a. calcitriol. Does anyone have the book with them? Out? No, don't worry about it. Dr. Sarkar and Dr. Kays have a bit of a discrepancy over which one's vitamin D3. According to Dr. Kays, it's cholecalciferol. According to Dr. Sarkar, it's 125. I think I want to confirm with your book that it says the same thing. Yes. I don't believe that at all. No? But I have no idea about that. I think we would know about it if he did. I think because colleges are like, we have a Nobel Prize winning teacher here, and I haven't heard about a single thing. Like, that's a huge thing. Like, universities like Berkeley have a couple of Nobel Prize laureates. No, I'm not going to say it on the podcast. I don't think that happened. So, things you need to know. You need to know the enzymes. You need to know the rate-limiting enzyme, 1-alpha-hydroxylase, and you need to know that this happens at both the skin, the liver, and the kidneys. This is a three-tissue process, which means that if you have deficiency in any one of these, you have problems with vitamin D synthesis. The most important is the kidneys. If you lose the kidneys, you are deficient in vitamin D, flat out. You lose the kidneys, you're deficient in vitamin D because it supplies the rate-limiting enzyme. The active form is calcitriol. It's triol because it's 1,25-dihydroxy, so there's two of your alls, and then it's cholecalciferol. So there's three alls. It's calcitriol. It has three alcohol or three hydroxyl groups on it. Calcitriol. That's supposedly why it's vitamin D3 referencing the three all positions, but I don't know that. I think the consensus was, when I looked it up, cholecalciferol is vitamin D3, but I don't remember now. Okay, so cholecalciferol, vitamin D3, that's what you're going to see when you take vitamin D3. This is what you're taking, because you guys have all, I'm sure, seen vitamin D3. You take cholecalciferol, and then it's converted by your endogenous enzymes. So if your kidney is failing, are you going to take is taking vitamin D3 going to cure vitamin D deficiency symptoms? No, because you don't have these enzymes anyways. Now, 25-hydroxycholecalciferol, which is not the active form, can also be 24-hydroxylase to 24-25-hydroxycholecalciferol, where it's stored in the liver. So, we make cholecalciferol, Then we hydroxylate it at the 25th position. We get 25-hydroxycholecalciferol. We can 24-hydroxylase it to store it as 24,25-hydroxycholecalciferol, dihydroxycholecalciferol in the liver. Or we can take 25-hydroxycholecalciferol. I can pronounce all this just fine. 25-hydroxycholecalciferol, we can 1-alpha-hydroxylase it to make 1,25-dihydroxycholecalciferol, aka calcitriol, which is the active form of vitamin D. So you need to know, rate-limiting enzyme, kidney, 1-alpha-hydroxylase. The active form of vitamin D is calcitriol for the 3 alcohol groups, or 1,25-cholecalciferol, or dihydroxycholecalciferol. Please know those. That chart is really important. If you memorize that chart, you're doing good for vitamin D. Both the skin, the liver, and the kidneys are involved in vitamin D synthesis. Production of calcitriol is regulated by a negative feedback mechanism. So, parathyroid hormone, you guys talked about that yet in physio? Parathyroid hormone activates 1-alpha-hydroxylase. Parathyroid hormone says your serum levels of calcium are low. Parathyroid hormone says your serum levels of calcium are low. If your serum levels of calcium are low, it's going to stimulate the kidneys to make 1-alpha-hydroxylase, which is going to synthesize active vitamin D. Because the primary thing that active vitamin D does is, vitamin D increases calcium absorption. Vitamin D increases calcium absorption, and it actually stops calcium excretion. So if parathyroid hormone says your calcium levels in your blood are low, it makes the thing that reabsorbs calcium. It stimulates 1-alpha-hydroxylase to make vitamin D, so calcium can be absorbed from the gut, reabsorbed from the kidneys, so your calcium levels go up. So parathyroid hormone, it directly regulates vitamin D synthesis because it acts on the rate-limiting enzyme. Vitamin D also increases phosphorus absorption. Vitamin D does two things. It increases calcium absorption and it increases phosphorus absorption. So, if either calcium levels decrease or phosphorus levels decrease, so a decrease in serum calcium or a decrease in serum phosphate stimulates the synthesis. Now, all three of these things, these are 3 regulators of 1-alpha-hydroxylase. The 3 regulators of 1-alpha-hydroxylase are parathyroid hormone, low calcium, low phosphorus. All 3 of those will activate 1-alpha-hydroxylase, which makes vitamin D, so you reabsorb calcium and you reabsorb phosphorus. You're going to get hammered on this in physio, so know this stuff, own this stuff. It's important stuff, too. It's a big thing for patient care. Because phosphate and calcium, what are those importantly involved in? Where do we keep calcium? Bones. So these are big things. These are big things in stuff like osteoporosis and osteomalacia and rickets and all that stuff. Any bone diseases, parathyroid and calcium. You're going to talk about a disease called hyperparathyroidism. Hyperparathyroidism, actually, what parathyroid does is parathyroid says you're low on calcium. So one thing parathyroid hormone does is it activates vitamin D to reabsorb calcium. Guess what the other thing is? It takes calcium out of storage to raise the blood level of calcium. Where do we store calcium? Bones. So if you have low intake of calcium, or if you have hyperparathyroidism, you're going to take calcium and take calcium and take calcium from your bones, and eventually you're going to have deficiently calcified bones, and you have major problems and fractures and stuff like that. So, 3 regulators of vitamin D synthesis. They regulate the rate-limiting enzyme. Parathyroid hormone, low calcium, low phosphate. All of which are calling for increased calcium or increased phosphate because the mechanism of action of vitamin D is to reabsorb calcium and absorb phosphate. Higher amounts of skin pigmentation by melanin decrease vitamin D synthesis. Why? Skin pigmentation by melanin, what does that mean? You're tan. You're dark. You have darker skin. Decreased vitamin D synthesis. Why? It stops the sunlight from hitting the 7-dehydrocholesterol in your skin because that's what melanin does. Is melanin absorbs UV radiation. It's there as a protective mechanism. So actually getting tan is protecting you from sunlight, which if you're not tan and you spend a day out in the sun without sunblock and you sit next to a tan friend that spends the day out in the sun with sunblock, you're like, oh, great, thanks. I'm a lobster and you turn to brown. But melanin is a protective mechanism. It protects you from sunlight, but it also protects the enzyme, or I'm sorry, it protects the substrate, 7-dehydrocholesterol, from sunlight as well. So the darker you are, the more sunlight you need. In other words, if you are of African descent, you need more than 15 minutes a day of sunlight, I'm sorry to say. It competes with 7-dehydrocholesterol in the skin for photons. Thus a black person with dark skin pigmentation requires about 10 times the exposure as a white person. Very politically correct, just saying. Okay, functions. It regulates calcium and phosphorus absorption. Vitamin D increases calcium and increases phosphorus absorption. It decreases calcium excretion because it wants more calcium. And it indirectly regulates calcium levels in the bone. Sure, cool. The more calcium you have, the more calcium your bones have, I guess. Parathyroid hormone increases calcitriol synthesis because parathyroid hormone says our calcium levels are low. Parathyroid hormone says our calcium levels are low. So we secrete parathyroid hormone when our calcium levels are low, and when our calcium levels are low, what do we want to do? Raise calcium. How do we raise calcium? We activate 1-alpha hydroxylase, which makes calcitriol, which reabsorbs calcium. Parathyroid hormone says that. Calcitonin is the exact opposite. Calcitonin says our calcium levels are too high. Calcitonin and parathyroid are like insulin and glucagon. Calcitonin says our calcium's too low, give me calcium. It takes it from the bones, it makes vitamin D to reabsorb it, and it gets more calcium. Calcitonin, I hope I said that right, that was parathyroid. Calcitonin says our calcium levels are high. I'm going to pack calcium onto the bones, and I'm going to stop vitamin D synthesis because I don't want any more calcium. They work exactly the same way that insulin and glucagon do, except they're talking about calcium instead of blood glucose. Questions on that? These guys are important. You got a whole chapter on them in physio, and nutrition and all that stuff, so know it. Biochem ties in heavily with those classes if you hadn't gotten every time I say physio, nutrition, and all that stuff. So, deficiency of vitamin D, hypocalcemia, right? It reabsorbs calcium. So, if you don't have vitamin D, you have low calcium. If you don't have vitamin D, you have low phosphate, because vitamin D also regulates phosphate reabsorption. Easy. Defective skeletal mineralization. Why? You have no calcium, no phosphorus, you can't mineralize bones. It's important. Vitamin D regulates calcium and phosphorus reabsorption. You have no calcium, no phosphorus, you can't mineralize bones, so defective skeletal mineralization, which means that those two disease conditions of defective skeletal mineralization are rickets and osteomalacia. Rickets is in kids. It causes bowed legs, deformed legs, enlarged head, enlarged joints, etc., etc. You'll talk more about it in radiology. And in osteomalacia, in adults, it's the same thing. They're soft bones is what the malatias are, soft bones. In kids, we call them rickets. And again, it's because you don't have calcium, you don't have phosphorus, you can't mineralize your bones. You get the collagen, you get none of the hard stuff. Food sources, don't care. Oils, milks, eggs, livers. RDA, I don't remember him telling us to know this one. At-risk population for deficiency conditions, elderly. Because A, their synthesis goes down, B, their kidneys might be failing, and C, they don't go outside. All three of which. 2, shut-ins. They don't go outside, they don't make the first step. Doesn't matter if you have the rate-limiting enzyme if you don't turn 7-dehydrocholesterol into cholecalciferol, aka vitamin D3. Or those with impaired fat absorption, because it is a fat-soluble vitamin. If you don't have fat absorption, you never pick up ergosterol. You never pick up 7-dehydrocholesterol, so you can't have those to make vitamin D in the first place. Medicinal uses, osteoporosis, psoriasis, those have both basically all been proven wrong by science, and don't worry about it. You might want to know a couple of those, but they're kind of general. You need sunlight, so if you're shut in, if you're old, if you don't have fat absorption, you're going to be deficient. If you don't have fat absorption, you're going to be deficient in A, D, E, and K. And vitamin D in large doses is toxic. 100 milligrams a day or greater for months can lead to hypercalcemia. I think it says at least mine said hyperkalemia. Hyperkalemia has to do with potassium. Hypercalcemia has to do with vitamin D. You can get calcium deposits in tissues. If you have high calcium, you get what's called dystrophic calcification, which is just calcification of soft tissues. So it's bad. Now, I know a lot of people that take more than 100 milligrams a day of vitamin D, especially in the wintertime when the sun goes away, to keep their immune system strong and all that stuff. And I've never seen this, but this is the supposed mechanism. Too much vitamin D, too much calcium absorption, too much calcium, your body does what it can with it. It gets calcitonin active because calcitonin says we have too much calcium, and it starts putting it places. It puts it in bones. It puts it in fat. It puts it in joints and tendons. You get calcification of this stuff. If you guys have ever heard of calcification of tendons or fats or stuff like that, this is one of the supposed mechanisms of it. All right, two more. We're blowing through this not nearly as fast as I thought we would. But you guys are good. You're rolling. I think the next two are short. So really important, physio, clinical nutrition, all that stuff. And Sarkar. You guys good? Okay. Vitamin E, TQ, most important natural antioxidant. What vitamin keeps vitamin E replenished as an antioxidant? Vitamin C. Vitamin C antioxidizes vitamin E. Remember that. So vitamin E is an antioxidant. Vitamin C fixes vitamin E. So now we've talked about three of our antioxidants. We're not going to talk about selenium until the minerals chapter later. But A, C, and E are our three big antioxidants. Selenium is important too. It's effective at high oxygen concentration. That's why we did ACEs. Selenium and E are at the bottom. It's good at high oxygen concentration. Mostly found in plant and animal oils. Hooray, it's in an oil. It's a fat-soluble vitamin. Active fat absorption promotes vitamin E absorption. Malabsorption, you get deficiency conditions. We've talked about all that. It's transported by lipoproteins, which are incorporated in the chylomicron. Fat-soluble vitamins, fat transport, chylomicrons, liver, all that stuff, LDL and VLDL. It is stored in the liver 5 to 6 months, TQ, stored in the liver. That is weird, because that is not what the front of the chapter said. The front of the chapter said A, D, and K were stored in the liver, and E is stored in fats. I object. I have no idea which one it is. I don't think it's a big thing. It might be. Ask him that when you get here in class, which fat-soluble vitamins are stored where, and if you find this in your textbook, because that's what you guys all have, is that textbook that he sells in the library. See where it says the fat-soluble vitamins are stored, and make sure you confirm where it is, because that one of those might come up on the test, where the storage locations of fat-soluble vitamins. It is effective at high oxygen concentration because this book says so, and because it's at the bottom of ACEs, and ACEs goes from low to high oxygen concentration. It is the first line of defense against oxidation, especially in fatty acids. Oh, here we go. Selenium metabolism. Selenium is one of your other antioxidants. It's an essential component of the enzyme glutathione peroxidase. It's a second line of defense against peroxidases. So just in case he asks, vitamin E first, selenium second. Both are active at high oxygen concentrations, though. This is not really part of vitamin E. This is just a side note. Selenium is another, it's a micromineral. I think we'll talk about it in a couple of chapters. It's important for antioxidant capabilities as well. It's good for high oxygen concentrations, and it's a second line of defense behind vitamin E. So vitamin E is first, selenium is second, and vitamin C keeps vitamin E going. So, vitamin E also is important for these things, TQ. Maintains membrane permeability. How does it do that? It incorporates into the membrane and it changes the permeability. It's a fat. It can incorporate into the phospholipid bilayer. It changes the permeability because it's bent. Bent stuff makes it harder to pack it together. It increases permeability. You guys kind of remember me talking about that? Polyunsaturated fats or unsaturated fats have a kink in them. When they try and line up in that little phospholipid bilayer, they don't lay straight. They're rebels. They're your girlfriend or boyfriend who lays sideways, or your dog, because dogs are infamous for this, that lay sideways on the bed, so you can't get 3 people anymore. You can't even get 2. You get 1 and a tiny dog. And that's what these guys are doing. They've got a kink in them. They're not laying straight. When they don't lay straight, the membranes have gaps in them, and they increase permeability. They're important in fertility control, and they improve vitamin A absorption. So 3 things you got to know. Membrane permeability, fertility control, and vitamin A absorption, and I guess that's part of it here. Make sure you know they're antioxidants. Vitamin E is a primary antioxidant. Vitamin E is a primary antioxidant, but it also does this other little crud. Vitamin E deficiency. Guess what? You're going to get free radical damage. Why? Because they're not being antioxidized. Free radicals are one of the primary things you antioxidize. Free radicals are damaging cells that have extra electrons. They're oxidized molecules. So what does E do? It goes and rips that electron away. It says, screw you, you don't get that, and it takes it away. So if you don't have antioxidants, you get lots of free radicals. If you have lots of free radicals, you get lots of free radical damage. Because those guys with one electron love to go around and tear shit up. So, vitamin E deficiency, just like everything else, malabsorption, etc., etc. Requirement for vitamin E is increased with polyunsaturated fats. But free radical damage, you can get anemia of the newborn, which isn't really explained how that has a mechanism of action. I don't really know. I apologize. But causes decreased hemoglobin, shortened RBC lifespan. I wouldn't worry about that too much, aside from the fact that vitamin E may cause anemia of the newborn. I need you to commit that to memory in case that's a TQ, but he's not going to go into the mechanism of action on that one, to my knowledge, unless he hits it hard in class, which he certainly didn't for us. Just anemia of the newborn, vitamin E. And lastly, neurological disorders, because inefficient fat absorption as well as vitamin E absorption. What's really important for your nervous system? Nervous system? Fat, right? Your brain's like 70% fat. What surrounds all of your neurons? Myelin. Myelin is fat. So if you don't have an antioxidant that protects that fat, your nervous system goes to hell. Your brain, your myelin, all that stuff needs antioxidants. Vitamin E is the primary protector of the fat in your body. And that's another thing I should talk about. These guys tend to work on what they can interact with. Vitamin C is water soluble, so vitamin C will antioxidize water soluble stuff. Selenium, I believe, is polar since it has a charge to it. Glutathione peroxidase is a protein. It has a charge to it. It will tend to antioxidize water-soluble stuff. Vitamins A and E are fat-soluble. They will tend to antioxidize fat-soluble stuff. So vitamins A and E are really good at protecting against fat-free radicals, which is why they say if you take fish oils, you need to be taking vitamin E, because you're taking a massive dose of oils that have great free radical potential, especially because they're unsaturated. Remember, unsaturated oils are more prone to produce free radicals. Never mind, clinical nutrition. Unsaturated fats are more prone to produce free radicals. So if you're taking something like fish oil, you need to be taking a fat antioxidant, which is why usually if a doctor prescribes it, they should, or if a good doctor prescribes it, they give you fish oils and vitamin E. And a lot of them come together now. Fish oils plus vitamin E, you'll see them in the same dose. Like your fish oil will have vitamin E in it. Because they know that to keep that thing protected, to keep it from going free radical and from going rancid in the container, they give it vitamin E, which protects it from damage. So vitamin E and vitamin A, since they're fat-soluble, will tend to work on fats as antioxidants. Selenium and C tend to work on water-soluble stuff as antioxidants. So, neurological disorders because of the importance of fat, free radical damage, and anemias. Food sources, fantastic, don't care. Susceptible to destruction by oxygen, heat, metals, and light. Just like everything else, keep it in the fridge, keep it in the dark. It's destroyed by commercial cooking and food processing, including deep freezing. All the stuff that you normally do with food destroys vitamin E, just like we talked about. 10 milligrams a day, 8 milligrams a day for women. Again, ask him about the RDAs on these bad boys. I don't remember. I thought he said we needed to know all of them for the fat-soluble vitamins, but I don't remember. It was a little while ago. At-risk population, premature infants for hemolytic anemia, those with fat malabsorption syndrome. If they don't have fat absorption, they have fat malabsorption. They have no vitamin E. If they have no vitamin E, they're at risk of anemia. And vitamin E toxicity, most unlikely, you need 50,000 IUs a day. That's a lot of vitamin E. I think the vitamin E I have at home is like 1,000 IUs a day, and that's a big one. So you need to take like 50 capsules of vitamin E every day. I think it's for a long period of time, too. And guess what? Increased risk of hemorrhage and increased blood coagulation time. Ooh, scary. So you need to take this for a long time to see any effect, and even if you saw an effect, it's probably only going to be a testing effect, not a clinical effect. You might. I don't recommend you dose patients up on it, but just for fun. All right, last one. Ah, crap. Page and a half. We got this. Get out here a little early. You guys ready? You want to fly? No? You want to learn it? Okay. Vitamin K. Two sources. Phyloquinones found in plants and menaquinones found in animal sources. It's easy to remember. Men are animals. And phyl has something to do with plants. Think of chlorophyll and stuff like that. Please know that phyloquinones are found in plants. Menaquinones are found in animal tissues. Those are the two sources of vitamin K that we get. Menaquinones and phyloquinones. You're just going to have to memorize quinone. Or just realize that he's probably not going to trick you with anything else, and you vaguely got some inkling that there's a Q in it, and I need to know men are animals and phyl is plants. And that it has to do with vitamin K. Very important. It has to do with vitamin K. Because he will throw stuff like that on there. He will say, what's the precursor of vitamin K? Menaquinone, phyloquinone, or the animal precursor of vitamin K. Menaquinone, phyloquinone, gluonolactone, or something else. He'll throw stuff from other vitamins on there. So you need to recognize that gluonolactone is vitamin C, phyloquinones and menaquinones are vitamin K. K and Q kind of sound similar, right? K, Q, something like that. The active form, which doesn't really say on here, is vitamin KH2. Vitamin KH2. Is that reduced or oxidized? Bam, nice job. That is a reduced form of vitamin K. The reduced form of vitamin K is the active form. It also says up here, intestinal bacteria are able to synthesize it. Intestinal bacteria are able to synthesize it, so please make sure you know that. All your intestinal bacteria are very important. Absorbed in the small intestines in the presence of bile salts, requires normal fat absorption. It's incorporated into chylomicrons, and it's accumulated in the liver. Over time, its storage is limited in the liver. So, guess what? Normal fat absorption, malabsorption, chylomicrons, all the same stuff I've been saying for every fat-soluble vitamin. I'm done saying. Because this is the last fat-soluble This is the last fat-soluble vitamin. I got to enunciate my words. TQ, TQ, TQ. Blood clotting, vitamin K. If you see blood clotting, vitamin K. Or vitamin E toxicity, but that's not going to be a TQ. Vitamin K is very essential for blood clotting. It is involved with clotting factors 2, 7, 9, and 10, TQ. 2, 7, 9, and 10. Vitamin K is involved with clotting factors 2, 7, 9, and 10. It also acts as a cofactor of gamma carboxylase, which converts glutamate to gamma carboxyglutamate. And this is how it works. So, it takes glutamate, which is an amino acid residue in these clotting factors, and it turns it into gamma carboxyglutamate. When vitamin K cofactors gamma carboxylase and turns it into gamma carboxyglutamate, it increases the calcium binding potential, TQ. It increases the calcium binding potential, which allows these cofactors to bind calcium. When they bind calcium, they are active. So, clotting factors 2, 7, 9, and 10 are calcium dependent. They have to have calcium. They can't have calcium unless the enzyme gamma carboxylase can carboxylate glutamate to increase its calcium binding potential. So they're calcium dependent, and indirectly they're vitamin K dependent. TQ indirectly. This was another one I missed on the test, I think, that year. Vitamins 2, 7, 9, and 10 are calcium dependent. True. They're directly calcium dependent. They're indirectly vitamin K dependent. Vitamin K has no direct effect on 2, 7, 9, and 10. But it activates an enzyme that activates that increases the calcium binding potential of 2, 7, 9, and 10. So look for the key word, direct or indirect. Vitamin K has an indirect effect on the binding potential of these guys because it activates the enzyme that increases calcium binding potential. Cool? So it activates gamma carboxylase, which turns glutamate into gamma carboxyglutamate, which enhances the calcium binding potential for 2, 7, 9, and 10. Now, not quite there yet. Prothrombin contains 10 residues of glutamate. I don't think that's going to show up on your test. I don't think he really goes through this. Okay. Yeah, he doesn't really spend a lot of time on that. If he does, tell me, and I'll go back and work on it. But there's a lot of classes that cover the clotting cascade, and he's not one of them. He just kind of blows through it and then doesn't spend a lot of time on it. What you do want to get from this chart, you don't need to memorize it, you're going to have to know it eventually, is that every factors, are they even on here? They're on there somewhere. Factors 2, 7, 9, and 10, which are on there, are both calcium and vitamin K dependent. To be calcium dependent, they must be vitamin K dependent. Calcium and vitamin K work together for the clotting cascade. Calcium and vitamin K work together for the clotting cascade. The one thing he might ask you is that the last step takes prothrombin to thrombin. And thrombin will take fibrinogen to fibrin. Fibrin is what forms the clot. Fibrin is a fibrous net that catches red blood cells, catches white blood cells, and forms a clot. So, after you get this whole clotting cascade where vitamin K activates gamma carboxylase, which increases calcium binding potential of 2, 7, 9, and 10, which allows 2, 7, 9, and 10 to activate, the end result of this huge cascade of steps in here is that prothrombin turns into thrombin, and thrombin converts fibrinogen to fibrin. Remember, pro and ogen are inactive forms, so we get these cleavages to make them active. Fibrinogen goes to fibrin. Fibrin is a fibrous net that starts to catch stuff, red blood cells, white blood cells, and forms a clot. That's why we get a clotting cascade. All you need to get out of that, 2, 7, 9, and 10, calcium dependent, vitamin K dependent, indirect effect on gamma carboxylase. Indirect effect on gamma carboxylase. I think that was the question. God knows how I remember these. I don't remember the other 48, but vitamin K has a direct or indirect effect on gamma carboxylase to increase calcium binding potential in clotting factors 2, 7, 9, and 10 or something like that. But make sure you know it's an indirect effect. Cool. The active form of vitamin K, here you go, is vitamin KH2, which is the reduced form. We have the vitamin K cycle. Okay, so vitamin K is inactive. It is reduced to KH2. It is reduced to KH2, which is the active form. And then KH2 is the cofactor for gamma carboxylase. During the process of gamma carboxylase converting glutamate to carboxyglutamate, vitamin K goes from KH2 to K-epoxide, which that's a little bond. K greater than 0 is actually the epoxide representation of a bond. K-epoxide. K-epoxide is then turned back into vitamin K, and vitamin K is then turned back into KH2. KH2 is the active form. KH2, since it's the active form, is the cofactor for gamma carboxylase. KH2 is the cofactor. During the process, he probably won't ask this, but just in case, during the process of it being a cofactor for gamma carboxylase, it's converted to K-epoxide, and then epoxide is converted back to vitamin K, and K is converted back to KH2. But KH2 is the active form. Vitamin KH2 is the cofactor for gamma carboxylase. For 2, 7, 9, and 10. Questions? Last little bit. You guys done? Almost. Don't leave yet. I see y'all packing your bag. You're not. I'm just lying to the people on the podcast to make you sound worse. Vitamin K deficiency caused by fat malabsorption or excessive antibiotic use. Why excessive antibiotic use? Because it's formed by gut bacteria. Vitamin K deficiency can cause a hemorrhagic disease, not just in the newborn, in anyone. Why? Why? Why can it cause a hemorrhagic disease? Because it does blood clotting. Fantastic, because it does blood clotting. It does 2, 7, 9, and 10, which are intrinsic to the blood clotting cascade. So it can cause hemorrhagic disease. Potential for food drug associated with anti-clotting drugs. In other words, if you take anti-clotting drugs, don't take vitamin K, because vitamin K is a clotter. I don't suggest you take Coumadin for any reason whatsoever. But if you're taking Coumadin, you don't want to be taking vitamin K because they have antithetical effects. Food sources: dark green leafy vegetables, spinach, cabbage, and liver. Ooh, I didn't know liver was a dark leafy green vegetable. But apparently it is. So we're going to go with that. Spinach, cabbage, and liver. Your salads can be upgraded. You need it daily. It's resistant to cooking, 60 to 80 micrograms, and megadoses of vitamins A and E antagonase. Megadoses of any fat-soluble vitamin are going to antagonize each other because they all compete for fat absorption. You need bile salts. You need chylomicrons. If you take a lot of one of them, you're going to decrease the absorption of the others because it's going to balance it's going to force the other ones out. And this is cool. High doses of the natural form, the plant form, phylloquinone, has no toxic effects. High doses of menaquinone, or menadione, I guess, can be toxic. This is the case with pretty much all vitamins. Iron, you can't get iron toxicity from eating too much spinach. You can get iron toxicity from taking too many iron supplements. When we make these things, especially as animals, we store it in a way that's meant to be stored. When we synthesize these things for vitamins, what do? We maximize absorption. So we force it into the body, both when it's stored in us and when it's synthesized for a supplement. So you can get overdoses of all of these things if you take them or if you eat animal products that have them, because that's what we're doing. We're trying to absorb them. If it's a plant product, our body only absorbs what it needs. It literally chooses, I'm done with iron. I don't need any more iron from spinach. It's awesome, which is another reason why you should get most of your vitamins and minerals from plant sources, or from, natural plant sources. Okay, that's it. There's one more thing I was going to say. Oh, this is a controversial thing. If you've ever heard of hemorrhagic disease in the newborn, you ever heard of them giving newborns vitamin K shots? It's because they're worried the mom is deficient in vitamin K, so it's pretty much standard procedure, unless you can talk the hospital out of it, that they will stick your kid, your newborn, with a needle and inject vitamin K. In case the mom was deficient, if the mom was deficient, the kid is also deficient, the kid might have a hemorrhagic disease, so we're just going to, as opposed to testing the mom, prior to pregnancy, we're just going to stick the kid with vitamin K. I personally don't agree. My personal opinion, I don't want my newborn stuck with a needle with vitamin K if whoever's having my baby is not vitamin K deficient. That's dumb. But that's one of the big things out there right now, along with antibiotics and erythromycin in the eyes, is that one of the standard things for a newborn is they will stick them with a vitamin K injection in case they're vitamin K deficient so they don't hemorrhage. Yeah. That's not ultimately up to me. That's a choice you get to make, but that's a choice I have to talk someone else into. So we'll see about that. That's where I'm leaning. The more and more I learn about it, that's definitely where I'm leaning. Hell, we did it for 5,000, 10,000 years without hospitals. Okay, that's it. You guys get out of here. If you have any questions, come talk to me, or we can continue this conversation off podcast where they're not going to fire me. Thank you guys for being here. I appreciate it. And I will see you before.